nice to see those gross pieces of, male and female repro stuff. Hopefully for the other labs, the lab sessions, you guys will also be able to see some of the other technology stuff that is normally very, very cool. And this is the kind of stuff that is, again, widely used, in practice, in actual companies and so on. So that's the nice part of the class. Now, returning to the even more fun side of this class, which is our class room, kind of. We're going to move now to establishment of pregnancy. So in the last class, we finished the estrous cycle and puberty. And we spent quite a bit of time, not only even away from the slides, going over how some of the estrous cycle thing works. I think at this point, you guys should have a much better, understanding of, what it was, some of the anatomical stuff, but also some of the physiology stuff that is tied with it. So all the way from follicles to sperm cells. And we went on to look at, did we go over fertilization? We didn't. Okay, so let's go to this first. So, but the overall speaking, the side of it is like, once we go over the fertilization side, which I really thought we were already here. We're not going to go. We're going to go fertilization. I apologize for it. Nice. So I thought we had gone through those already. So sorry. So the idea is that once we finished that part and we went over extensively on the estrous cycle, now it's the time that we know more or less, we completed all the basic stuff of the sperm cell. Then we completed all the way to ovulation. Okay, so we do know, from a pretty good, standpoint, what it takes for the body to produce, the male body to produce a fertile sperm cell, and what it takes for the female body to then produce an egg. Yes, obviously much more complicated on the female side, but now what we're going to do is talk about a little bit more on the physiology of fertilization, and now we're going to start to talk a little bit more also on the synchrony that needs to happen in order for this to occur. So let's start now from both points of view, and we will start from the sperm side of it. And on the sperm cell side of it, a lot of what we're going to do, it has to do with obviously the transport of that sperm cell through the female reproductive tract. So all the way from being deposited in the vaginal canal to moving through the cervix, moving on through the uterus, going to the ovduct, spending some time in the ovduct, and eventually when the stimulus is correct, being released in the ovduct and then swimming all the way through the oocyte in order to fertilize. A few things that we're going to talk, and now we're going to be able to tie it up with some of the, especially when you go back and see the anatomical parts and how actually the sperm cell is built, you will see that there's going to be a few major things happening. One, during this transport, you're going to see that by the time the sperm cell is actually deposited in the vaginal canal, from that point on, there's one thing that happens, which is that's a foreign body inside of the female reproductive tract. So lots of the stuff that happens inside of on top of some of the biochemical stuff that is kind of very specific for fertilization, the female tract doesn't necessarily know the sperm cell is there. So all the way from the pH of the vaginal canal to macrophages and neutrophils, you're going to try to trap and kill sperm cells. Okay, that's what the female body will try to do. Obviously, on the other side, the sperm cells will have, some things on their side to go as fast as they can all the way to the oviduct. Why? Because in the oviduct is a kind of a site where they have somewhat of a safe site to be, deposited, and then from then fertilization will occur. So let's start with some of the stuff of the sperm transport. Site of deposition, for the most part, vaginal canal. You will have some species that you can have actually the sperm cell being deposited inside the cervix, but those are not very common. So overall, as a general rule, is in the vaginal canal. Secretions, all these things, immobilize sperm cells within 1 or 2 hours. Rapid transport is essential, and this kind of, it has to be fast because it is not a very good environment for the sperm cell to be in, okay? Lots of the stuff that we talked about at the beginning about, the role of the male sexual glands, which is to add volume, to add nutrients, buffers, minerals, and so on, is going to be heavily used at this point because this is really going to be the vehicle where, some of the sperm cells will be actually transported. So if you think about some of those things, these are like kind of common sense. Once you understand that the sperm cell is actually a foreign body, you will try to see, yes, it has to move fast. Do you remember way back then I told you about the sperm cell is this little alien, it has the tail, it has to swim fast, but at the same time I said, it is not going to be through that tail that they are going to move all the way from the cervix to the oviduct. So you guys saw, some of the tracts from bovine. Okay, so you saw the horn of the uterus, and that, depending on the tract, is a little smaller, a little bigger, but let's say all the way to the oviduct, you have something like this in terms of length. A cell swimming through this length here, it's like marathon, okay, for them. It's too much. So most of the time, or not most of the time, all the time, what's going to take from the cervix all the way to the end of the uterus is contraction from the female tract, okay? So you will have contractions on the direction going from the cervix to the oviduct that will actually push the semen and the sperm cells all the way through. How fast? How long do you think it takes for the sperm cell in those tracts that you guys saw in the lab to go from the cervix to the oviduct? Just a guess. 2 hours, 3 hours, 1 day. How long do you think it takes? I tell you 10 minutes. Is that fast? Yeah. That's about it. That's 10 to 15 minutes. That's how long it takes. So are these 10 to 15 minutes important for the development and actually the maturation of that sperm cell? It will be. Because you're going to understand that, at the same time that those seminal plasma is kind of important to protect and to give the vehicle for which they go, one of the things that nature does some wonderful things and some very strange things at the same time, removing some of that seminal plasma from the environment, the surroundings of the sperm cell, will also be necessary for that sperm cell to modify, especially the surface of the cytoplasm. The surface of the head and the cytoplasm will have to be modified. And one thing that we were going to be later talking about. So yes, rapid transport, contractile activity of the myometrium. So you're going to have And the endometrium, they have this ability to have contractions not only towards the oviduct, but imagine during parturition, the contraction has to be in the other side. Go ahead, sorry.
Question with the previous slide. What do you mean by advantage to A?
Oh yeah, advantage to A, sorry. So the thing with the artificial insemination, and this is not artificial intelligence, but it's artificial insemination, it is that most of the time you're going to deposit the semen inside the uterus. It's not in the vaginal canal, it's not in the cervix, it's actually inside the uterus, which basically means you can skip 2 barriers during this process. One of the reasons and one of the advantages for that is like, one, you're going to have probably, proportionally speaking to the amount of sperm cells that you're depositing, more going towards. And the second part is that you can use a lot less sperm cells in order for fertilization to occur. So I think, in the beginning, and we're going to have some of that in one of our labs too, but normally we, artificial insemination gun is like a little pipe like this size, very, very narrow, and that you have to palpate the cervix and insert through the cervix that thing, and inside of that you're going to have a little straw, and this straw is going to have a certain number of sperm cells. In this case, in the industry standard is something like 25 million cells. But compared to what you have to use for during a regular breeding and ejaculation from male is a few billion cells. So this is one of the advantages, like from a perspective of using this commercially, you can have one ejaculate from a bull, for example, and separate it into dozens of doses, and you can use that same one. So you dilute into a very, very specific kind of media, and then you can dilute and so this is one of the reasons. Okay, very good. So reservoirs, this is the other stuff that we mentioned before, that the cervix, then the utero-tubal junction that we talk, so that little kind of space, or bottleneck between the end of the uterus, the tip of the uterus, and the actual oviduct, those will serve as reservoirs, which means you're going to have a continuous influx of sperm cells going in, either the uterus or the oviduct. And actually the oviduct can be considered also a site of deposit, a certain deposit of sperm cells. All of them will have, particularly before reaching the oviduct, the problem will be that those are hostile environments. They can be around, there being stored, but they're gonna be killed eventually. So they cannot stay there for much longer. But the oviduct tends to be a little better. They have a specific site for them to be there. But you will see that as a rule of thumb, your sperm cell will be viable inside the reproductive tract of the female for about 1 day. That's overall speaking for most species. Yes, you're gonna have exceptions, yes. Dog, canine species is one of them. They can survive for a lot longer than that. So this is what they call, so some of these reservoirs, so you're gonna have this kind of, slow release, continuous release of the sperm cells. Part of this is to avoid having too many sperm cells reaching the egg at the same time. It's kind of, it's paradoxical. You have a few billion cells in an ejaculate and you have only one egg. So you're gonna try to end for that egg. It's not like it is, is it okay that just one fertilized? It's not that it's okay, it has to be only one. So there has to be a certain number of sperm cells that reach the egg in order for the egg to avoid what we call polyspermy, which is fertilization by more than one sperm cell. And the egg has a very, very nice mechanism to avoid that. But if you have, 1,000 sperm cells reaching the egg at the same time, the egg might be difficult, but if they have just 100, they might be able to do what they have to do. So far, so good? Yeah? Nice. So, opposite direction of transport, yeah, this is going to happen in the oviduct because you're going to have the egg going this way from the ovary, the sperm cell going this way from the uterus, okay? Some of the things that happen is that by this time, by the time the egg is released, the sperm cell will already be in the oviduct waiting. And then eventually when ovulation occurs, there is a trigger that will make them go in the opposite direction. So the oviduct also actually has this opposite movement or direction that they can sustain, in which we call peristalsis and anti-peristalsis. This is the same kind of movement from a physiological point of view that your intestines will have and your esophagus will have, so it's not very different. Again, very, very, very complex nervous control. Survival, we talked about this. Lots of this is because of physical barriers and phagocytosis. We mentioned this. The COC, this sometimes is the acronym for cumulus oophorus complex, which is basically, you can understand, it's the egg. It's the whole thing. It's the egg plus a few cells that is attached to it, which we call the cumulus oophorus, that will be actually released with the egg during ovulation. So, two words in this slide that will be key for you to understand fertilization: capacitation and acrosome reaction. Okay, those two things. One of which, the capacitation, is this overall scheme or this overall reaction by which some glycoproteins in the head of the sperm cell will change. Are these important? Yes. If you don't have that change, the head of the sperm cell do not bind with the egg. If they don't bind, in order for the next reaction to occur, which is the acrosome reaction, if that binding doesn't happen, this acrosome reaction doesn't happen, it will not happen in the right place, which is in the zona pellucida of the egg. That's the place where the egg will bind. Then you're going to do the acrosome reaction. The head will then penetrate the zona pellucida to reach the plasma or the plasma membrane of the egg. And then once the plasma membrane of the egg meets the plasma, the remaining of the plasma membrane of the sperm cell, is when the two membranes can fuse and the nuclear envelope of the male can enter the cytoplasm of the egg, which is the overall or the ultimate goal of the sperm cell. Okay? Now, all this maturation, and I think we went too far here, maturation of the epididymis and stuff. Yes, you are at some point, this acquisition of motility. Way back then in one of our first classes, I told you that by the time the sperm cell leaves the seminiferous tubule, it's not exactly fertile. Okay? It's not ready to fertilize. One of the reasons is because it's very immotile. It cannot move. It will acquire that as it matures along the epididymis. By the time it's in the coda of the epididymis, which really is the big storage, before ejaculation, that's when it acquires that. Does that mean that we'll start, by the time it reaches the female tract, that little alien starts moving like crazy? Not necessarily, okay? The body or nature made a few changes there because at the end of the day, there is not that much energy inside that sperm cell, even if it's surrounded. So if it starts moving too early, by the time they really needs to move, they won't have enough energy. So what the body does is okay, in 15 minutes, it goes with the flow, the contractions of the female tract is deposited at some point in the oviduct and stays there. And by the time you will see that by the time it is in the oviduct, they have kind of a hypomotility. There is, things that when they are there during capacitation, they're not moving a lot. And then after fertilization, or not after ovulation occurs, they have the right stimulus to start going in what we call hypermotility state. So the motility, it is important. We understand why it's important. I think one thing that is nice for you to understand, they have to move at the right time. They cannot be moving around like crazy. This capacitation, so it starts really, by the time it starts moving through the female tract, and you start having all that seminal plasma being removed from the environment where they are, and they have to rub through the female tract, that already is the start of capacitation. How long does capacitation last? Like most of the capacitation will occur in the oviduct. Why? Because really they don't spend that much time elsewhere. Okay. And it will take something like 6 to 8 hours. They have to be, and this is an important step to understand, for example, why we do what we do when we use certain biotechnologies for reproduction. The idea that you have to have those sperm cells spending 6 to 8 hours before in the female tract actually is necessary, or this is actually super important for you to time when you breed the cow. So if you're going to do, for example, artificial insemination, you have to account for some of that. You have to really synchronize when ovulation will occur. That basically means that if I expect an animal to ovulate at a certain time tomorrow morning, it means that I have to inseminate this cow or do artificial insemination sometime before. And for that, you have to take into account that they have to spend some time in the female tract to be optimal, for fertilization. Acrosomal reaction tends to be a much more straightforward thing because it will start to happen really by the time the egg or the sperm cell really meets the zona pellucida. Why? What is the zona pellucida made of? Is it made from cells? Remember the egg, you have the egg, the cytoplasm, the nucleus, but you have that layer around the egg. Yes or no? No? Sort of, more or less. Okay, so it has this layer. Okay, we're gonna draw. So this layer is kind of a glycoprotein layer that surrounds, the egg, and it is a big barrier. Okay, it is a super barrier, but it is an absolutely necessary barrier. For once, if you didn't have that barrier, you would have free access from the sperm cells to reach the egg. And with that, basically, I would say, unless you have a lone survivor that just reaches the egg, which is very unlikely. Think about it. There was 1 billion cells, somewhere in there. If there is only one, which is unlikely, then you have a chance to have a normal offspring. Otherwise, you're going to have, an egg with, having several, sperm heads or sperm nuclear heads going inside, and you're going to have genetic problems, okay? So let's go with so this is epididymal maturation. That's not too much. It's just a slide or a table with some of the stuff that happens, during the maturation of the sperm cell. But let's go with this, which is a much more important part, which is capacitation. So it is absolutely necessary for sperm egg to fuse. It needs somewhere about 6 to 8 hours. So you will see that for some of the technologies we use, we don't have to care so much about the capacitation, but we have to understand that once we start to manipulate, semen and sperm cells, we have to account for this. So if we do something with the sperm cells, that starts triggering capacitation, it is a problem. If we do everything in vitro, like in vitro fertilization, either for humans or for animals, for some of the industries, such as the bovine, in vitro fertilization is a big deal now. So people produce a lot of embryos through in vitro fertilization. So if you do that, yes, you have to do something with those sperm cells to mimic capacitation. You're going to have to capacitate those sperm cells artificially. Okay? It's going to start in the uterus, and then it's going to, at some point meet the beginning of the ovduct, and that's where capacitation will mostly, where this is mostly going to happen. Okay, that early portion of the ovduct and a little bit through the uterus. Okay. Most of the stuff, and this is perhaps one, like if I ask you what is capacitation? Capacitation then is going to be, yeah, it's a process that is necessary for, sperm oocyte fusion, but is in reality what it is a modification of the surface of the sperm cell. When you say is a modification of the surface, yes, you will have modifications in the cytoplasm in order for the surface to change. But what you end up doing, you're modifying the amount and the types of glycoproteins. We're not going to go into details of which glycoproteins and so on, but you're changing the surface of that sperm cell. Okay? And once you change the surface of that sperm cell, it means that by the end of capacitation, by the time capacitation is done, this sperm cell will be able to attach to specific receptors in that zona pellucida of the egg. Okay? Some of these things here, cholesterol efflux, protein kinase dependent phosphorylation, or modifications of the mitochondria, basically this phospholipid layer is stabilized. This is the last bullet there is basically the step before acrosome reaction occurs. Okay, so you're going to need to have that in order for, if you imagine and if you remember the head of the sperm cell, you have that acrosome vesicle. That acrosome vesicle is filled with, very potent enzymes. You're going to have to get rid of those enzymes that will basically digest tissue. So what you have to do during capacitation is, yes, you have to prepare the membrane to attach to the zona pellucida, but you're going to have to make that sperm cell head a little weaker. So by the time, when I say, and this is what the destabilization of the membrane means, and that means that, once it attaches, it will basically dissolve some of the cytoplasm, and all those enzymes will go in. We're gonna go into more detail. Okay, so it's basically modification, removal of stuff from the surface of the sperm cell in order for binding, okay, for binding to occur and to prepare for the next one, which is the acrosome reaction, which is the activation of the acrosome. Another set of little things, factors controlling capacitation. We're going to go through this as one of the slides. Every species will have somewhat of a different factors that control capacitation. Okay, all of them will have like little things that can trigger. One of the things, let's start with the very first one, which is physical. The fact that when the sperm cell will have to go through the cervix and the uterus, you're going to see like this in here, you call this thing like this, kind of a rubbed off, all the stuff that is around the sperm cell is removed. Lots of the seminal plasma is removed. This is one of the stimulus, okay? here is saying, what is like a nice one that is supposed to be capacitated, a sad sperm cell here that, eventually may not be capacitated the way it should. But it has to do a lot of this, like this cervical mucus, scrubbing. So you're removing some of the stuff. You have the effect of some reactive oxygen species. Oh, but does that make sense? Yes, you're going to have a few, cells, not only defense cells around there that will have some reactive oxygen species around, but even the media, the cervix media, the mucus, they have some of that for protection. But that, in turn, is actually used for the sperm cell for it. Basically, what happens here is that you do have some of that. You are changing, for example, some of this cholesterol here. And when we say before cholesterol, normally you have quite a bit around. By the time it moves through the uterus and the oviduct, you have a lot less. So when you say efflux, it's like removal of some of that cholesterol from the head of the sperm cell. You start adding some other things. Among those things, some of this sialic acid binding protein and some of these receptors. So you have a few more things. Some other species, progesterone tends to have somewhat of an effect, even the molecule on the head. If this is not done more or less the right way or in the correct order, you can have, I have one of those points there that says you have been prematurely capacitated. And this can actually happen. And if it's prematurely capacitated, if it is not capacitated at all, or is prematurely capacitated, that basically means that by the time it reaches the egg, it's not going to be ready. Or it was ready before. And by that time, it's already either dead, or it won't be able to perform fertilization as it should. And then by the time it reaches, this is the oocyte. These are some of the cells of the cumulus ophorus. So it's going to have to swim through it. And you see, like this guy here, by the time he reaches this stage, he has to be at a point where he's moving very fast. He has the right combination of glycoproteins and some other receptors in the head, and he can actually bind the zona pellucida. Okay? So for this slide, if you say efflux of cholesterol, cervical mucus scrubbing, sialic acid binding, and over here you can have like calcium influx to allow hyperactivation, it's already enough. Okay, so those are key points that you say, oh, Ronaldo, but I read in some other paper that, for whatever, sperm cells of whales, that's different. Yes, it's going to be different in some species than others, but at least you have this overall view that you have to, change the surface. Does that make sense so far? Yeah? No? That's good. Acrosome reaction. So acrosome reaction tends to be, as I said, straightforward. By the time it was there and it reached, the sperm cell reached the oocyte, I said, has to be the right combination of receptors and glycoproteins attached to the zona pellucida. And as I said, the zona pellucida will basically be this, this glycoprotein layer. So yes, you will have the little guy here. It has the right combination of glycoproteins. Happy. Looking up. Okay. To the zona pellucida. And the zona pellucida, if I have to draw the egg, and if this is the size of the sperm cell, it would be like even bigger, like enormous. Okay. And this will have to bind to the zona pellucida. The zona pellucida has this, you're going to see later, for the male, the female, what we call zona pellucida proteins. Okay, so they call ZP, and sometimes they call ZP1, ZP2, ZP3. So it's zona pellucida 1, 2, 3. They have a few types. And it has to bind with some of them. How important is those changes in the surface that was caused by capacitation? And how important is those ZP? One example, people have tried to do contraceptive methods by creating antibodies against some of these zona pellucida proteins. If you kill some of those proteins, or if you kill those proteins, in some way or another, or you make it difficult for the binding to happen, yes, it can go all the way to make this animal, for example, sterile, not having offsprings at all, not allowing fertilization. This is one of the methods. So when this occurs, and then remember, like if you go here to the head, you have the head, you have the plasma membrane, and you have like the nucleus here, and you have the little helmet from the gut. Okay? This is the acrosome vesicle. What you're trying to do is to put this guy out of here. Okay? So once let's say the first step was, okay, you met here with the right receptor, attaches. Well, just attaching won't make it to the actual, this is the zona pellucida, but this is the actual cytoplasmic membrane of the egg. Sometimes you can read in books, they call it oolema. Okay, this is the specific name of a cytoplasmic membrane of an egg. This guy here, or portions of this cytoplasmic membrane of the sperm cell, have to fuse with this. Meaning, this guy here, you have to find a way to penetrate through it. Okay? And the way to penetrate through this is the acrosome reaction. Because inside of this acrosome, there are like very potent enzymes that will then digest this. Okay? And will make what? The head of the sperm cell to go inside. Oh, Ronaldo, but what do you mean? So you're saying that this thing will have to be freed. So are you going to destroy the actual membrane of the sperm cell? Yeah, a portion, not all of it, but a portion of that will be destroyed. So once you fuse these two here, you're going to poke holes so this thing here can flow outward. And when I say outward here, it's towards the zona pellucida. Okay? So these hydrolytic enzymes, we say hyaluronidase and acrosin are two examples of these very potent enzymes that will basically digest the zona pellucida, will allow the head of the sperm cell to enter the egg or through it, and then eventually a portion of the head will actually fuse with the cytoplasm of the egg. Then you're going to have the actual nucleus inside. By the time it's inside here, you will know that we call the male pronuclei, and you're going to have somewhere here the female pronuclei. Okay? That's the moment where you turn a haploid cell, which is the egg, into a diploid cell. Okay? The moment when the two chromatins get together and reorganize, what you call syngamy. Okay? And once that thing happens and you have the complete set of the DNA, you then call that cell a zygote. Okay? few more tables for you to read about the acrosome reaction. It's nice, but all of those things, having that idea of the acrosome reaction is enough for us. It's always good to have a few more things. And this diagram is basically, overall speaking, is this. You're going to have to go through this. We explained already here. You're going to release the contents of the acrosome content or the acrosome membrane. You're going to go inside and say, Ronaldo, what the hell is this that this would be the actual zona pellucida. This is the actual cytoplasm. You see that eventually you have to fuse the two somewhere in here, and you're going to put inside the nucleus or the pronuclei of the male. What the hell is this, Ronaldo? This little cortical granules. You didn't explain that in that one. So what is that? That is a mechanism that in the next slide I'm going to explain that is called basically is the mechanism by which the egg avoids polyspermy. Okay? And I'll explain in detail how that's done. Okay? Oh, sorry. So this, basically speaking, in this case, is very simple. Once you actually have let's say you have the head of the sperm cell is here, and it did go through and it fused with the female. By the way, you're never going to have everything of the sperm cell inside. You're not going to have the entire sperm cell entering, the oocyte. It is just the head that then frees up the nuclei, and it goes. Once this happens, there is a lot of influx of calcium towards the inside of the cytoplasm. Every time there is influx of calcium for any cell, something will happen. You will understand that all of these second messenger pathways eventually depend on calcium for something to happen, from either transport of stuff inside the cell to actually activation of second messenger mechanisms. So you will have this guy here entering, and you're going to have lots of calcium, either coming from outside or from the endoplasmic reticulum of the cell. Okay, so you're going to have lots of calcium. Once you have calcium inside, one of the things that happens is that you have these little structures in the periphery here of this egg that they call it cortical granules. Okay, so yes, they are granules that is in the cortex of the oocyte. Okay, so they're there. They're not activated, they stay there. Once that happens, and once fertilization actually happens, and this influx of calcium go in, you start transporting those cortical granules to the actual membrane, towards the actual membrane. And once they are transported here, they fuse with the plasma membrane, and they deposit whatever that is here in this space. You see this, when I say this space, it is this, you have the zona pellucida here, you have the egg, the actual cytoplasm of the egg here. You have this space, okay? It's a very tiny space, between the two. But it's an important space. Why? Because once you deposit those cortical granules into that space, what you do, there are few things that happen. One, you have a physical, you put them apart from a physical standpoint. So this is much wider, that gap. Now, the other one is that you harden that gap, and you also harden the zona pellucida with that cortical granule. Basically meaning that, if the first one entered, and let's say these are a few other ones that are trying to enter. Once this one entered and this gap here became like white, these ones here can no longer deposit their nuclei inside of the egg. And this is how, you know, you avoid this process that we call polyspermy. Okay? It has to be only one. That's it. And that's how it avoids. Now you understand why, if you go back, why do you have some of the storage sites? Why do you have a little less sperm cells reaching the actual egg? You cannot have that many. Like we've done research trying to collect, for example, embryos, where we could see how many heads of sperm cells were here. And you can have all the way from 2 or 3 to over 100. But you never go much beyond that. Basically meaning that, you know, although you have a few billion cells being deposited on the ejaculate, you're not going to have that many reaching the oocyte. Very good. Well, you got the idea here. Transport. So let's have a 5-minute break, and then we'll continue with the egg transport. Hold the wrist.
Someone from her. I think it's.
Okay, that's fine.
It's like video tissue and then it can. I'm still everyone. I was surprised. Yeah, and I know a lot about it. So I haven't found that so far, but I'm still tweaking. So once I finalize everything, yeah. Like, bigger. Yeah. So like, that's just you have the previous.
What is that?
Do you have the?
Yeah. Yeah. Very good. So let's continue here. So I actually end up already obviously talking a little bit about the ovum or the egg transport, but on the side of the egg, I think there was more complicated things happening before. You know, during the process of ovulation is a lot more complex on the male than on the male side, but basically speaking, the female or the egg, it's for once, it's what's going to determine is the most important piece over there in the synchrony for fertilization to occur. It lasts less time in terms, a viable sperm cell, as I said, can last up to 24 hours or so. An egg will last for 6 to 8 hours only, after ovulation. So it has to be fertilized somewhere in that range. Oh, there will be cases, yes, there will be cases where it can last longer and so on, but overall speaking, that's more or less the range. It will be after ovulation, as I said before, the egg will be, once it is released, it has the egg, obviously, which is with its zona pellucida, but over here you're going to have lots of those cells around the egg that we call the cumulus oophorus, and those cells will play some role, during the fertilization process. And actually, you have to have some of those cells around. Once we do, for example, collection of cell or eggs or whatever technology we want to use, one of the ways in which we classify the quality of the eggs is the morphology of these cumulus oophorus cells around the zona pellucida. Okay. Once the egg is released, or once ovulation is released, I said during the anatomy part of the class that you have the ovary. For most of the species, the actual the ovulation does occur in the cortex of the ovary because most of the structures are in the cortex of the ovary. And eventually, if ovulation occurs somewhere here, you will have to be picked up by the oviduct. And you have that initial portion of the oviduct that we call infundibulum. And they have like this, let's say, very large opening, which is very fimbriated, basically meaning you have like hair-like projections from those cells. And what they do is like they start beating towards the inside of the oviduct. So it creates more or less of a negative pressure here where it sucks the egg towards the inside of the oviduct. Okay? As I said, it releases the cumulus cells and the egg. You do have some of the fluid from the inside of the follicle actually going inside as well. They will be somewhat important too. And you're going to have lots of movement at that point. You're going to see that, obviously after that, you're going to have lots of transformation in the ovary because that ovulated follicle will then slowly be transformed into the corpus luteum, okay? And in the other side, you're starting to have the movement of the egg towards the center of the oviduct. Hopefully, at that point, you do have sperm cells already capacitated and waiting for this to happen. And one of the things that will happen, and once this egg was released, what was the main driver or what was the main hormonal stimulus for ovulation to occur? What is it? What needs to happen to trigger ovulation? LH. So what needs to happen to LH? A surge of LH. So once the surge of LH occurs, the actual sperm cells that are capacitated and waiting actually are released from the crypts of the oviduct, and they trigger that LH surge also triggers that hypermotility pattern that the sperm cells will then acquire. So it is a very synchronized thing. So theoretically, within a few or very fast after ovulation, you're going to have the encounter of sperm cells with the egg. And again, at that point, I said peristaltic, anti-peristalsis movement, one going this way, another going this way inside of the ovule, okay? Like transport time, and when I say the transport, time, this is basically the transport. This is not how long it takes for fertilization. Maybe I should have put this in a different way. This is not how long it takes from ovulation to fertilization to occur. This is actually the amount of time that it takes for the egg to leave the oviduct. During this time, fertilization have occurred, if fertilization were to occur, okay? Yes, can you have, so does that mean that every so many days, whatever that is, that your estrous cycle lasts, even if you're not fertilized and you're not gonna get, pregnant, are you going to have a little egg in the uterus? Yes, you will. An unfertilized egg, but you will have. What happened to that egg? Well, probably your defense cells will take care of it, okay? Or something else, or you're gonna go die pretty quickly. So it's a one-cell thing. But does that mean that you're going to have that thing going? Yes, you will, okay? Fertile life of the egg, 12 to 24 hours. Most of the time less than that, more like 6 to 12, in many species. Oh, is it important, Ronaldo, that 6 to 12 or 12 to 24 and so on? For the sake of the class, I think it is just interesting to understand that it's like very short. It lasts for not very long. From an industry or from an applied point of view, it is very important because this is what you use. The same way that you use capacitation and how long a sperm cell can last, you're going to have to understand how long these eggs or these oocytes can last in the female tract or after ovulation. Because that's where you synchronize your breeding. In a normal situation where you have the bulls and you don't have to control or manipulate the estrous cycle, nature has done that for us. You understand that once the female accepts the male, everything else will be more or less lined up from a hormonal and process point of view that the fertilization can occur. But once you have to do that artificially, you're going to have to control yourself. Humans will have to say, oh, for that sow or for that cow or for that mare, I will have to wait for estrus to occur so I can predict when ovulation will occur? Or do I go and give, for example, an injection or of some hormone that triggers ovulation, and then I'll have to know that from injection, how long it takes for ovulation to occur, And from that prediction, I can say, okay, so if this is how long it takes, I have to go back a few 8, 12 hours and say, oh, this is when I'm going to inseminate. Okay? So it brings a whole lot of, logistics around. And does it mean that, that happens for those that has experience in taking, for example, pills as a contraceptive method, people or doctors, they emphasize very well that you cannot miss. You have to take it every day and you cannot miss. For some of these manipulations of the estrous cycle, it's the same thing. You cannot miss. If you miss a certain injection or if you miss the timing that you inseminate, that's it. That's all it takes for fertilization not to occur. Okay. Some of these things here in terms of the transport of the egg, it's pretty straightforward. Ovulation occurs, picked up by the infundibulum of the ovduct. It travels towards the sperm cell that is hopefully capacitated, and then fertilization will then occur. Okay. What does it that we mentioned before? I think this about the cytoplasmic and nuclear maturation. At that point, the cytoplasmic maturation is almost complete, meaning everything's accumulated over there. Nutrients, DNA, RNA, is all there. It's not DNA, but RNA is all there. But meiosis of the on the nucleus, it's then resumed after the L8 surge. So it has stopped up to a certain stage. When L8 surge occurs, it continues, and then eventually will only finish after that nuclei enters the cell. This is when the actual very, very last stage of meiosis on the female side will actually be done. So basically meaning, you do remember the polar body that you have, which is basically your remnant of the meiosis, process. Very good way to call your little brother that you don't like. You're a polar body. He will not understand, and you say, yeah, you're a remnant of a genetic material. So the stuff is, once that polar body will have to be expelled, really, more or less as fertilization occurs. Okay, so you have this, and this is the pronuclei that you have, and all the cytoplasm, and it's going to be very easy to see sometimes like a little thing here going towards actually this space. So you're going to have like your zona pellucida, your cytoplasm, and you have a little ball, in the, lost in there. This is the very last step of meiosis, which is to remove that DNA material that is there. Okay. Then, actually, when that occurs, this is when you're going to have the complete nuclear maturation. Then you have ready this guy to meet this, and you form a diploid cell, a diploid cell, which is then officially the zygote. Okay? This process of chromatin, as I mentioned before, is called syngamy, and then with that you form the zygote, which is the very first cell you will ever be. Okay? And after that, so at that point, this is and one of the reasons why also, from a stem cell research standpoint, this is where people like to work a lot with some of those embryos, not the zygote stage because it's kind of a short-lived, but all the way to morula or blastocyst, or I could explain later, because those cells are what we call pluripotent or totipotent cells. They can basically be differentiated into anything you want. So from a stem cell research standpoint, you'd like to have access to that kind of cell. Why? Because today you have ways to differentiate some of these cells into specific tissues. But if you have to have like a already specialized cell like in the case of Dolly. Way back then when they did cloning, they had a cell, if I'm not mistaken, from either mammary gland or skin. So you would have to do what? Get that nuclei that have been differentiated to become, let's say, a skin cell. Undifferentiate that cell and redifferentiate into something else. That's a big process. It's a very inefficient process. It's not very good. It takes a long time. It's much more costly. But that part, at least in the human side, the problem with that approach is what? Why people don't like to use embryos for research? Ethics. It has nothing to do with biology. It has to do with ethics. It has to do with, do we use embryos? In that case, yes, embryos that you can transfer, embryos that can give rise to another offspring in order to do research. In animals is a little bit of a less ethical barrier, but for humans tends to be a big one, okay? Very good. So in this fertilization here, as I said, male pronuclei, female pronuclei, put it together, fertilization, singamy occurs. Normally, it happens here in the center of the egg. You're going to have, we're not going to go through this, how the chromatin gets together, but you have the intermixing of the chromosomes first, then you have the zygote, and after that you have what we call the first cleavage division. At that point, all the way up to day 8 or 9, because by the time it's day 8 or 9, the embryo is still surrounded by the zona pellucida. It's only going to, and this is the word, the technical word that we use, is only going to hatch from the zona pellucida by about day 7 or 8 of existence of an embryo. Up to that point, what you do have, and now you're going to understand why cytoplasmic maturation was so important. You're going to have to do all of those things from the content of the egg, of the cytoplasm of the egg. Okay, there's not going to be much coming from the outside into the inside of the egg. And when we say cleavage division, is that cleavage division, Ronaldo? Yes, it is division, but you're not increasing in size. You're just getting this big ball, and splitting into 2. And then you split into 4. And you split into 8, 16, 32, 64. And then you go into an exponential progression, of divisions. By the time that thing goes into day 7 or day 5, day 6, day 7, then you have hundreds of cells. Up to that point, maybe day 5, day 6, they still look all the same. And after that is when you start to have differentiation of those cells. What do you mean by differentiation, Ronaldo? Long story short, that's when you're going to start slowly having those endoderm, ectoderm, mesoderm. You're going to start to have cells that will differentiate into the placenta, those cells that will differentiate into the embryo proper, the actual embryo. So all this, that single cell will have to give rise to all these tissues, okay? So the idea then on the first class or the next class probably, we're gonna go through a little bit of that. We're not gonna go into embryology, which is a class on its own, okay? That we're going to go, it's the ectoderm that gives rise to skin and neural tissue, the mesoderm that gives rise to muscles, bones, the endoderm that gives rise to this. No, we're not gonna, then you have somites, you have the notochord. If you go back to your drawers in your brain, you're gonna remember some of these words, but we're gonna skip through some of that, and we're gonna try to go straight to placenta, gestation, and parturition, okay? okay? Otherwise, we're gonna get stuck in that for another 10 classes, okay? So, why did I put parthenogenesis here or twinning? I think for us, obviously, this is not so much something that parthenogenesis will be super important for domestic animal species, okay? Yeah, that will occur in many others, but not for us. Twinning, on the other hand, is something that is, sometimes it is worth going through just because, you know, in some cases, it's not even twinning. Like the true twin is basically what we call the monozygotic twin, which basically means once this zygote is formed, theoretically, this zygote is to form one offspring. Okay, it's one. Somehow, that little thing in that first division splits, and they start giving rise to independent ones. This is what we call identical twins. Okay, so when they are born, they are like, yeah, one is the mirror of the other. Yeah, it's monozygotic twin. What is dizygotic twin? Hey, I have a sister. She doesn't look like me, and she is a twin. What is the difference? Is that my sister was born from a different egg all around, okay? From a different follicle. So basically my mom, I don't have a twin, but by the way, but it is just like this. What happens in this case is that you did have a double ovulation or a triple ovulation or whatever, okay? In the case of animals that gives of several offsprings, yeah, it's somewhat of a kind of twin, but it's more like a polyzygotic twins. You know, yeah, they're born from the same gestation, but they're not born from the same egg, okay? Is this common in other species? No, it's not, okay? So that would be the one that where you have true ones. Yeah.
For identical twins, the egg splits, does each egg still have like its own fertilization envelope, or?
No. It's basically like by the time this happens, because it has to be, it's not like you have several sperm cells and somehow this had supranuclei. It is just that in the first cleavage, and this is the part where we don't have that much understanding of that first stages. It's not that we don't have, but it's very, very like cloudy so far. Because imagine one thing, you have to have that cell and they start to go into cleavage. So for example, you can artificially go into that. For example, I think we have done this by the time they are like 32, 64 cells. Go with a scalpel and make it two. You can give rise to two offsprings by just you going there. Oh my God, no. Cut this in half, each one, that's it. So you can do that. So the cell at that point doesn't really understand it has to give rise to one offspring. Okay, so somehow during that process, naturally do that. Okay. Now, you can imagine that after this happens, you have to give, I said, these cells have to give rise to all tissues in your body. One thing, for example, that we do not understand well is like, how do the cells know that, first of all, some of them will become ectoderm, mesoderm, endoderm? How does some cells say, I'm going to become a placenta? How some of the cells say, once you are in the mesoderm, some of these cells might say, okay, I'm going to become the tip of the finger. And the other cell will say, you know what? I'm going to become a liver. Imagine how complex this is. Like today, if we knew that, we could maybe artificially create an offspring in lab, but today we can't. Like we can do lots of things in these early stages in terms of technology, and we can do very little after the thing is differentiated or after differentiation. When I say differentiation, having organs and systems and so on. here on Thursday.